Now we are coming to the next immunity, acquired immunity. Acquired immunity is also called a specific immunity. Now I'll take what is the meaning of acquired immunity. Now this immunity is developed or it is acquired. Acquired is received after the birth. As I told already, when the child is born, the child is having 20 to 25% of immunity. That is called a natural immunity. That is called an innate immunity. But when the child is growing after birth, little by little, one by one, new immunity resistance will be developed. Such immunity which are developed newly after the birth are called as acquired immunity. Developed or acquired after the birth. Immunity is for specific pathogen. When acquired immunity is developed, one immunity is meant for one particular pathogen. During this development, antibodies are produced by the individual. The antibody is a specific for one particular type of microorganism. That is called immunity is for specific pathogen. One example we can take, when the child is at the age of 5, first time affected by chickenpox viruses. Chickenpox viruses are entering. It is called the first infection. Immediately the body is producing antibody for chickenpox virus. So this antibody will kill only chickenpox virus, not any other viruses or bacteria. That is called immunity for specific pathogen. The unique features are, for this acquired immunity, some special features are there. Unique features. What are they? We have to see. Antigen specificity, that only I told here. One antibody will specifically kill one microorganism, one type of microorganism. Diversity. When at a time 3 or 4 or even more different type of microorganisms are entering, the antibody will identify which microorganism it has to kill and it has to destroy. That it will do. That is called diversity. Recognition of self and non-self molecules. The antibody produced by the individual's body, human body, can identify which are all the chemicals belong to its own body, which are all the chemicals not belonging to its own body, coming from outside. He can easily recognize. And memory, it is very, very important feature, memory. As I told us just before, at the age of 5, a child is the first time suffering from chickenpox. Antibodies are produced. The antibodies are meant for chickenpox virus. Okay, the chickenpox is cleared after 1 week or 10 days or 15 days. But the antibody is there in the body permanently and it will never forget what type of microorganism it has to kill. Now at the age of 50 or 55, once again the second time the chickenpox viruses are entering. The antibody produced at the age of 5 is ready to destroy the chickenpox viruses that enter at the age of 50 or 55. That is called the memory. Throughout the life, the antibodies are keeping in memory that what type of microorganism it has to destroy. That is about this unique feature. All these things are the unique features. Now we are coming to the concepts of acquired immunity. The acquired immunity is having so many concepts. What are they? Two major concepts we are going to study. One is called a cell-mediated immunity. Now we can put within bracket here CMI, calculator. That is abbreviation for this. CMI, cell-mediated immunity. What is the meaning of the cell-mediated immunity? We have to see. Now in the blood, 3 types of blood cells are there: RBC, WBC, and platelets. But in the WBC, 3 different type of WBCs are there. One is the T cells, another one is macrophages, the third type is natural killer cells. So 3 types of WBCs are there. They are stimulated when the microorganisms are entering. Any type of microorganism, when they are entering, the 3 cells are activated. Immediately the 3 cells are making the lymphocytes. Lymphocytes is another type of white blood cell. Lymphocytes to be stimulated and they are produced more in number. So lymphocytes are produced enormous in number. Once the lymphocytes are produced more, the lymphocytes will engulf the microorganism, swallow the microorganism and digest the pathogen. Microorganism will be digested. So the cell, white blood cell is a cell, lymphocyte. It is digesting, engulfing and digesting the microorganism. That is called a cell-mediated immunity. Second one, we are coming to antibody-mediated immunity. Within bracket you can write abbreviation AMI, antibody mediated immunity. Now in this type of immunity, two types of cells are involved, white blood cells are involved. B cells, T cells. Two types of white blood cells are involved. They are multiplying rapidly when the microorganisms enter. They produce one special chemical that is called an antibody. But the cells are not attacking the pathogen directly like previous one. They produce a new chemical, antibody, and this new chemical is destroying the pathogen. So only the chemical antibody is destroying the pathogen. Pathogen is antigen. So the antibody is destroying this antigen. So since it is a chemical, we are calling it as antibody mediated immunity. It is the second concept of acquired immunity. Now we are going to take about types of acquired immunity. There are two types of acquired immunity. Acquired immunity, I already told you, after the birth, the immunity is developed slowly one by one. In that first one is called active immunity, second one is passive immunity. Active immunity means the immunity developed by the individual's body and it will be there for permanently for a long time. Now passive immunity, what is this passive immunity? There are certain microorganisms, they are very strong here, they enter into the body, but our immune system cannot produce antibody. They cannot destroy the microorganism. That means what? Microorganism is more powerful. Our body cannot do anything with that. So our suffering will be more. At that time, passive immunity. We are taking ready-made antibody from outside. Synthetically prepared medicine. That is nothing but medicine. We are taking medicine which is entering into the body, killing the microorganism. Then the disease is cured and the chemical or medicine is coming out of the body within 1 day or 2 days. That is called passive. Medicine will never stay permanently in our body. That is passive. Now we take about active immunity. Active immunity is produced actively, very effectively by host immune system, human individual's immune system, our own immune system. That is the first one. Then it is produced due to contact with the pathogen. Pathogen should come and contact the body, inside the body. Then only it will be produced. Or by its antigen, chemical, poison chemical should enter. The third point is, it is a durable. Durable means it is there for lifelong or for a long period of time. And it is very effective in giving protection. Very effective. Okay. Now very strongly it is acting on the microorganism. Immunological memory is present. I already told you the memory of our immunity developed at the age of 4 or 5, but they are throughout our life. That is called a memory. The next one, booster effect on subsequent dose is possible. Sometimes our immune system is induced by vaccine. So our body will produce lot of antibody. But in that first they will give one mild dose. So our body is start preparing antibody. Then after some period of time, they will give the second dose which is slightly stronger. Then our body will produce more amount of antibody. So permanently will become resistant to that. That is called a booster effect. Strong effect after second time or third time of that microorganism. That is called contact of microorganism. That is booster effect will be there. The last one, immunity is effective only after a short period of time. As I told, first time when the dose is given or when the microorganisms are entering, that immunity will not be very effective. It is now only start preparing. So it will take some time, maybe 5 days, 6 days, 1 week or 10 days. After that only it will become more effective. So it is very short period and it will become less effective. Then passive immunity we have to take. It is quick immunity. Okay, but it is coming from outside. Passive immunity is received passively. Passively means from outside. And there is no active host participation. Host means our body. Our body will never participate. Just as simply swallowing the medicine is acting. Our blood, our bone or our glands and all, they are not at all acting anything. That is no active host participation. It is produced due to antibodies obtained from outside. Antibodies means synthetic chemical medicine obtained from outside. It is transient and less effective. Okay, it is a transient. Transient means it is immediately is acting, but less effective means that effect will be there only for a short period of time, maybe 1 day or 2 days. That is called a less effective. No memory. Because the medicine is not going to stay in our body, it is going to be eliminated from the body. So there is no memory at all. Subsequent dose is less effective. For example, the same type of disease coming again and again. We are getting medicine from the doctor and we are consuming again and again. So what will happen? The medicine is not going to act on. That is, subsequent dose is less effective. The immunity develops immediately. Now this type of passive immunity develops immediately because once the medicine is taken, medicine is aggressively acting on microorganism. So immunity develops immediately, but this immunity is not permanent. It is temporary, only for 1 day or 2 days or 3 days like that. It is a less, there is what? Short period of time. That is passive immunity. Now we are coming to types of immune response. Now the immune response, the response is given by our immune system. Now we have to know what is that immune system. Actually our body is having 10 different type of systems. Okay, human body is made up of 10 systems. Digestive, respiratory, circulatory, like that we can keep one list. But in that immune system will not be there. But the immune system is made up of some organs belonging to other systems. I'll give some examples. The bones belong to skeletal system, but bones also play important role in immunity. The second one is the blood. It belongs to circulatory system, but the blood is playing vital role in developing immunity. So blood is the second. Third one, some glands which are present in the chest, near the heart, thymus gland, lymph glands. All these things are the glands which belong to other organs, other systems, but they also play important role in immunity. So they are all joining together forming immune system. So how the immune system is responding, that we have to say. That response, only two types of responses we are going to study. That is, first is primary response. Okay, the beginning itself, how it is going to respond, that we have to say. It is a temporary or early, we can say early response. Now the primary response develops during first infection. Okay, first time microorganism enter into the body, immediately our body will not aggressively act on microorganism. Our body will take some time for antibody production. So it develops during first infection. Second, immune system learns to recognize the antigen. That means first the immune system, like as I told about blood, bone marrow, glands and all, first they will learn what type of chemical composition of the antigen. First it has to recognize. Then only it can produce antibody. And produce antibody by the lymphocytes. Lymphocytes are white blood cells. Then only it will produce. So it will take time. It will take time for learning. It will take time for production. So that it will take very slow process. It is a slow process and a short lived. The slow process may be very short lived, maybe 3 days, 4 days, 5 days. After that it will become more active. Now this is called the primary response. First initial response. Then secondary response we are going to study. Okay, what will be the subsequent response? We have to see. Now assume that first a few microorganisms enter. Okay, antibodies start produced. Then maybe after a period of time, maybe 1 week or 10 days or 1 month or 1 year, second time the infection is coming, developed when exposed to the same pathogen, same microorganism, second time. Next second time infection is there, but it may be after 1 week or after 1 year also. Then at that time only this secondary response will be developed. Lymphatic system. The lymphatic system means the lymph vessels, lymph node, everything. Lymphatic system produce more antibody immediately because already they learned how to produce. Very quickly the antibody will be produced in enormous quantity. When more quantity of antibodies are produced, they can immediately act on microorganism. Many plasma cells are white blood cells, are produced that produce more antibodies in 1 or 2 days. It won't take long time. Within 1 or 2 days, sufficient and enormous amount of antibody will be produced. It is also called the booster response. Very effective. Large amount of response it is giving. Very strong response it is giving. That is called the booster response. Now we are coming to the differences between primary immune response and secondary immune response. What we have studied previously, we have to consolidate it. Now first I will take primary immune response. It occurs as a result of primary contact with the antigen. First time the antigen or microorganism is contacting our body. At the time this will be developed. Antibody level reaches peak in 7 to 10 days. So antibody production will be there sufficient only after 7 to 10 days it will take. That means long time. So, next, prolonged period is required to establish immunity. That means 7 to 10 days, sometimes it will be more also. So, long time is required to prepare ourselves to face, that is what, to act against the microorganism. There is rapid decline in the antibody level. Now, the antibody level, rapid decline. Now that means what? It is not immediately produce more quantity, little by little. At the time, it is some antibodies are acting on microorganism. Sometimes it will be denatured. So up and down will be there. The level of antibody will be ups and downs will be there. So last one, it appears mainly in the lymph nodes and the spleen. Lymph nodes, just we have discussed. Now lymph glands. Lot of bulging portions are there in the lymph vessels, and also spleen. In the previous video class, I told you, spleen is a pad of tissue. It is a gland attached at the bottom of the stomach. It is also important for immune system. So, that is, it appears mainly in the lymph nodes and the spleen. Now, we are coming to secondary immune response. It is a fast response, booster response. We are coming. It occurs as a result of second and subsequent contact with the same antigen. Antigen means microorganism or foreign chemical. So, second time the microorganism enters. Very quickly the effect will be present. Antibody level reaches peak in 3 to 5 days. It won't take about 10 days only. It will take within 2-3 days. The antibody response will be present. It will reach the maximum level and it will try to attack the microorganism. It establishes immunity in short time. That means short period of time. This is the short period of time. It is enough. Antibody level remains high for long period of time. Once the second time it is produced, that immunity will be there for ever, long time. It appears mainly in the bone marrow. I already told you, bone marrow is a part of immune system, followed by the spleen and the lymph nodes. Okay, so these are all the secondary response. So that means in the first response only lymph nodes are involved, but in the secondary response bone marrows are involved, and also the spleen and the lymph nodes are involved. Many are involved. So the immunity developed by secondary response will be stronger immunity.